Assalamualaikum and a very good afternoon to the audiences here as well as the online participants. Today we have Dr. Lam Ming Quan, the Assistant Professor, Department of Biological Science, Faculty of Science, Universiti Tunku Abdul Rahman, Utah. He will be presenting on wearing many hats, a slash career in multi-omics research, genetic consultation and science columnist. Before Dr. Lam begins his presentation, I would like to invite our director, AP Dr. Wang Siok Mui, to give a brief introduction on our speaker today. Thank you.
Thank you, Aisha. Okay, it's my pleasure to introduce Assistant Professor Dr. Lam Ming Quan. Okay, Dr. Lam graduated from University Technology Malaysia with a PhD in bioscience. He possesses multidisciplinary expertise and interests, including bioinformatics, microbiology, food agriculture, and environmental biotechnology. As an experienced postdoc research fellow, he has been involved in several international collaborative research projects with the University of York, United Kingdom. Currently, he serves as an assistant professor in the Department of Biological Science, Faculty of Science at Universiti Tunku Abdul Rahman. Dr. Lam is also a Yayasan Khazanah Scholar and alumnus. As a young academic with a strong track record in both research publications and teaching, he is committed to advancing scientific knowledge. Okay, beyond academia, he is a genetic consultant at Nucleus Education, where he provides insights into genetic analysis and its application in healthcare and biotechnology. Additionally, he is a science columnist for the China Press Berhad, where he simplifies complex scientific concepts for the general public. Okay, the floor is yours, Dr. Lam.
All right. Good afternoon. Is that everyone can hear me? Not sure. Okay? All right. So I'll start now. All right. Good afternoon. So I'm Lam. As introduced, I'm from Utah. Okay. So today I'm going to give a talk about a slash career. If you know the word slash means, in simple means multitasking, doing many works at one time. Okay, and have different professions. So this is a bit brief introduction about my background. So I graduated from industrial biology and master in biotech and bioscience from UTM. In fact, I'm a product of UTM. Okay, all right. So currently work in Utah. So I was a Khazanah scholar and alumni until today. All right. So most of the time, Khazanah gave me a lot of opportunity. So I appreciate their works and helps in developing my career. And then some of other works as well as introduced by Dr. Wang just now. Okay, so let's begin. I will rather describe myself as a person who are doing many works, which is cross or multidisciplinary. So sometimes people might define themselves whether they are work in wet lab or dry lab. So I don't do so. Okay, I will say I'm half and half. Okay, so half wet lab means the normal one that you know, like DNA, RNA extractions, those microbiological work. I'm also involved in dry lab, in which developing codes, doing bioinformatics works. Okay, so here are my two major research areas: multi-omics and bioinformatics analysis of halophilic microbial community and their enzymes. We focus on industrial application, for example, food, health, and environment. Another major research area is actually deal with the insect, black soldier fly larvae, to produce a new protein source as animal feed, okay, or food ingredient. Okay, so my research begins here. All right, so back to say 7 or 8 years old back. So everything starts with mangrove. So Tanjung Piai National Park is our famous national park. It's southern tip of Johor. So I'm a Johorian as well. So it's a good place for you to go to have sightseeing, but not to go to play so much. A lot of mosquito. All right. Okay. Okay, what happens with mangrove is that mangrove is a diverse environment, very unique in Southeast Asia, because Malaysia has been ranked as number four has large area of mangrove. Okay, we should reserve the mangrove well. Why? Because there are a lot of pressures. Okay, for example, there you found a lot of metabolites and enzymes from those microorganisms, and mangrove can be considered as the largest carbon requisition place. Okay, where you recycle a lot of carbons, right? So this is overview of my research currently ongoing. Okay, so for example, I have rhinocellulose degradation for biofuel productions, which is also part of my PhD, and food waste degradation for bioactive peptide derivations, as well as currently one of my projects dealing with environmental health, which focus on microplastic degradations, as well as the natural pigments with bioactivities. Okay, so everything start with technology. As a biotech graduate, so I acquire skills from UK's related to all the omics mentioned here. For example, metagenomics, metatranscriptomics, and metaproteomics, which deal with whole populations of DNA, RNA, and protein respectively. So this is what we call culture independent methods. Why culture independent? Because most of the microbe, 99% of the microbe in this world cannot be cultured due to the limitations on the medium itself. So those those culturable microbes on plate, they are only represent around 1%. So this is what how we separate those samples into culture independent and dependent, right? Okay, so of course for culture dependent, you have genomic, transcriptomic, and proteomics to do with DNA, RNA, and protein of that particular microorganism or organisms. Okay, so these are all the omics I acquired during my PhD time. So it's time for me when I work to utilize them. All right, so let's proceed. I'll integrate all those omics in different research that I mentioned just now. Okay, so the first one. Okay, so Malaysia, we are chief producer of palm oil as well as their waste. Okay, we are chief producer. Okay, no matter palm oil or their waste, so we are the chief producer in the world. So we have a lot of empty fruit bunch. You can see from roadside. Okay, the lorry bring a lot of fruit bunch. Then we go to a palm oil factory to process the oil palm. Okay, all the residue, one of the major residue here we call it empty fruit bunch. Okay, so during that time, we have a lot of these waste, and then we have also started with mangrove research. So what we think actually is that why don't we mix these two samples together and see what happens? Okay, so we mix the empty root bunch together with the mangrove soil sample. You see a lot of black color inside the flask, and you do not know what things going on. Okay, so after observation for a certain time, we see some degradation from SEM. You see some of the things here. You see that this one is the original one, is very compact and nice structure. After being eaten by the microorganism, you see some of the things being degraded. All right, so a lot of enzyme activities were observed that time. So it arise some of the questions. For example, who are the one going to do that? What they did, and then how they did it. So to answer these questions, the way we use meta-omics to do. Okay, so first, metagenomics will tell you who are they in terms of microbial structure, microbial community. So beware of the words metagenomics because it can mean many things. For example, if you talk about 16S RNA metagenomic, means that particular regions of 16S of whole microbial populations. Okay, while when you talk about shotgun metagenomics, which means that all the genes, all the DNA in that particular populations. So this is the difference. So when you go to find some service, please be careful the word metagenomics. You can ask further. Okay, would you give me all the genes within the population? Will you give me only 16S genes for all the population? This helps you to differentiate the service when you go to bioinformatics. All right. So just the tips are for you to deal with the supplier. And then we discover from the result that we have a lot of unknown taxa. Okay, a lot of unknown taxa, a lot of unknown microorganisms in the samples when we culture the mangrove sample with the waste for many days. A lot of things unknowns. Okay, so most of the thing we know only up to maybe phylum or family level. Okay, all right. So with that, we proceed to metagenomics, metaproteomics, and so on. So now we know actually who are they who are doing the work, and a lot of them are unknown. Okay, next one, we actually check what are the RNA highly expressed by the mangrove microbiome. Okay, microbiome means the microbial populations. So we saw some of the uh, non-expected enzymes, and also some of the enzymes that never been found before to work to degrade lignocellulose, the waste. Okay, so this is in RNA. So we proceed to metaproteomics to check the protein form. Okay, if you know in molecular biology, the RNA can be expressed, but they are not necessary to form proteins. Okay, sometimes they're not necessary to form protein. So we proceed to metaproteome to check the real protein form after the RNA modification or RNA processing. Then we actually have two fractions of protein. First one, we call it bound fraction. Bound fraction is the protein or enzyme that bound to the waste itself. Okay, usually it's solid portions. The supernatant one, as you know, the centrifuge, the supernatant, okay, found in the protein found in supernatant. Surprisingly, most of the enzymes or proteins, they actually bounded to the substrate. They are not in the supernatant. So if your experimental design is only dealing with the supernatant, you won't find any proteins, not many proteins. Okay, so most of them will be bound to the substrate. Right, so this is mangrove microbiome work on lignocellulosic biomass. So compiling who are doing the work, how they are doing the work, what they produce. So actually we can integrate all the data from the meta-omics work. You can check synergistic actions, like for example, bacteria A, they might degrade certain parts. Bacteria B, they degrade certain parts of the waste. Then they give you all the pictures coming up. Okay, so how to form this picture require lots of bioinformatics skills and your scientific knowledge. Okay, so this actually help us to find some potential candidates for further enzyme cocktails development. Right. Okay, not only that, so toast meta-omics is refers to toast unculturable, cannot be culture ops. So we also do culture omics. The culture omics means here, toast microorganism which cannot be cultured previously, we try to culture them out by using the meta-omics data actually that we found. Well, we mainly focus on media formulation. For example, through their metabolic pathway analysis, we actually can know that most of the mangrove microorganisms, they don't utilize simple sugars such as glucose. Okay, and then they require some vitamin, for example, vitamin B complex like biotin, to actually support their growth. So with this kind of information from meta-omics data, we try to culture those previously unculturable to culture. So we have a list of microorganisms, okay, those previously unculturable, and we culture it out and we utilize their genomes to perform the subsequent works. Okay, so one of the bacteria we name it as, we publish as a new species, is recognized by International Committee of Prokaryotes. So it's a new species that define here. If you search this name, you'll find my names. So it's called Robertkocha solimangrovi. Okay, it's named by us. Solimangrovi means isolated from the mangrove soil. Okay, I also learned some of the linguistic based on this, and also some of the environmental and biodiversity law due to this. Okay, we have more stricter law now due to ministry want to utilize this as our potential biodiversity resources. So we have a stricter law now. You need to submit the application when you want to deposit your strand in the culture center. Okay. All right, so that's about lignocellulose degradation. That's my 3 years PhD. That was my 3 years PhD. So now I'm dealing with a lot of food waste and environmental health. So let's proceed with the second one, food waste degradation for bioactive peptide derivations. So have you heard about soy sauce from Angel brand? You find it everywhere in the supermarket, even in Lotus. In Eon, you'll find this brand of soy sauce. Very common. Is produced by Malaysia. It's from Bidor Guangheng at Perak. So the only waste they produce from these factories is what they call it soy sauce cake. It's very salty. Okay, very salty means they actually need to settle this waste. So I have two directions for this food waste degradation. First one, we collaborate with them, use black soldier fly larvae to eat them. So to check whether the larvae can grow better for new protein sources. There's another story lah, but today is used for the other purpose. Okay, this soy sauce cake we try to degrade it, and because the soy sauce contains a lot of good proteins, for example, glycine, in which breakdown of them can release a lot of bioactive peptides. So we try to convert the waste to wealth so that those bioactive peptide could be utilized as potential food ingredient. Okay, so we perform the work. For example, we try to find some halophas, those can tolerate the salinity of the soy sauce cake, so that they can grow and degrade them and release some of the bioactive peptides that in the subsequent slide. So this is what we found during the transcriptomic data. Most of the proteolytic genes, they are upregulated, which means they produce much more higher production as compared to the control, which has no soy sauce cake. So we suspect these all of the proteins, they actually upregulated, not only upregulated, but they perform the work as to degrade the SSC, the soy sauce cake. All right, so after proteolytic degradations, we actually do some in silico analysis in which we simulate the digestions of enzymes and to check after enzyme digestions, what are the potential bioactive peptide produced. And we use certain software and server, for example, to check whether can be absorbed by human intestine, whether they are allergens or not, or non-allergens, whether they are toxic or non-toxic. So we check using the server. Is predicted lah. Okay, it's predicted by bioinformatics. So there are some of them can be represented here. Is that some of them, they are non-toxic and non-allergen, as well as can be absorbed by human intestine. It's predicted by bioinformatics. And then some of the peptides, they have function, for example, anti-diabetic functions and antioxidant functions. We tested in wet lab. So these are some of the peptides we found that we plan to further investigate to purify them, right, to check their properties later. Okay, that's about food waste degradation. So next one is about environmental health. I think during COVID time, we wear face masks every day until today. Although most of us did not wear today, but it's still important because we have a lot of disease, okay, airborne disease. We still need to wear the face mask. So what happened is that do you know that in Malaysia, we actually, our marine quality index is not up to the average level. So the government actually tried to push this up by providing more grants and more creative and innovative solutions. So why? Because the face mask is one of the pollutants, okay, due to this. So I even wrote an article for that. So to make an awareness related to face mask degradation. So with this casual chat with my supervisor and my friends, we started with the face mask degradation from especially using the halophilic bacteria, salt-tolerant bacteria from the marine environment. So why face mask is very toxic? Because of their structure. Their structures, the monomer of their structure, of course, is carbon C. So this carbon can be utilized by the microorganisms to become their nutrients. So we try to do this, okay, through the metagenome and genomics way. So with this so-called background, you know what we did is just go to buy some face mask and grind it and put in flask and put my mangrove soil together and check whether it works. Okay, as simple as that. So for several weeks, we observed that some of the weight loss of the face mask. Weight loss means some of the insoluble structure that has been degraded by the microorganism to form soluble carbon. Okay, so which means that something is working. Okay, and we isolate some of the strain out and check they are also halogen, okay, observed. All right, so and currently we are doing metagenomic analysis for this project. Okay, not only from the plate, we also see in SEM that there are some cracks and pits after degradations, and also bacteria attached to the surface of face mask. All right, so that's actually somehow it's proved that it should working on the face mask. Okay, several strands, we sequence their genomes, okay, and then after we search, we use some database to search for plastic degrading or microplastic degrading genes. We found there are lots. Okay, there are lots of potential microplastic degrading genes, especially this strand, CO35. This strand has not been explored with their application. They only have their taxonomy name in the database. So it's one of the rare bacterium file that they can degrade the microplastics. So we visualize some of the proteins using the most updated tools. We call it AlphaFold, in which the developer of AlphaFold won the Nobel Prize Award last year. Okay, so it means that the importance of the bioinformatics nowadays, as well as not only do the bioinformatics works, we also perform some of the FTIR analysis to check where the bonds are broken down. Okay, so CH bond tolls are related to the phase mass structure. So it's indirectly proof that they are doing some work. The microbes are doing some work on that. Okay, so with all the omics career starting 2017 until today, so there are a lot of thinking in my mind that what am I doing? What I'm doing? I was always in front of a computer like this, like this, dealing with database, server, software, hardware, and programming. So, and many people in biological science, they do not know the differentiation between these five. They do not know the difference between database, server, software, and so on. So most of our biological students, they thought that they are the same. They are the same, just word difference. No, there are differences among all of these. Okay, so what happens in front of my computer is actually a lot of coding coming out. A lot of the codes that need to resolve. Sometimes, as a biological students who learn bioinformatics is really tough, but I think it's workable. It's very encouraging nowadays to step into that area because it's important for us for molecular work as well. Okay, so this is what my daily life. If you see me in Starbucks or Coffee Bean, I usually doing these kind of things with a coffee. Okay, this code is wrong lah, that code is correct lah. Okay, that happens a lot, right? So those who are from IT, then they will know. Okay, so what happens in omics and bioinformatics currently is a lot of problems actually arise. Let me tell you one by one. First one is actually related to data quality and standardization. As you know, you extract DNA and RNA, you can 100 ways to do that. And you can send for many service using many platforms. Even one prediction of protein can use different software or tools. So there's no standard until today, no matter in which country or in which research. As long as you find certain mathematical equations, as well as those two who are works, you can do it. But most of us don't aware like whether the predictions can be wrong or not. So this happens a lot nowadays. This is one of the problems. We need to have a further improvement to check the data quality and standardize the process. Okay. Another one is, have you heard about bioinformatics? All are predictive data and inferred data. They are not being proved in wet lab if you don't perform so. For example, I can model a lot of proteins every day, but those proteins, whether they are really look like that, does not necessarily mean the true things. Okay, so those omics data, only the raw omics data you get from the Illumina or from the Oxford Nanopore, the raw data is not prediction. But all the data comes after that, they are actually inferred or predictive data. So those data can be wrong. So usually I told myself, don't trust that data too much because it can be updated. For example, like your phone, you have apps, right? You constantly update your apps when the Google tell you to do so. Okay, so same with bioinformatics data, they can be updated every day. So sometimes you see your bacterial genomes, oh, today I have 3,000 genes, but tomorrow might change. Okay, so this is what we encounter every day. So what I told myself is usually that, okay, at that time, the data is correct, then I follow that time. That's it. Okay, then with that, I carry on with my investigation. So this is what happens nowadays. Quite a common problem. Another one is data upcycling. So usually in publication, you see a lot of people publishing meta-omics data. They just focus on certain parts of genes. Okay, they don't utilize every single data. For example, in one genome, you have 3,000 protein coding genes. You might not utilize them in one publication. So what we're trying to do now is we try to upcycle the data, use back the data we sequenced previously to check whether those bacteria has another applications or not. This is called data upcycling, and you save your money. One of the important things is save your money. And then another thing is you don't create rubbish, a lot of rubbish here. So data upcycling is one of the potential problem. Okay, I think another problem is important for postgraduate students is that we lack of interdisciplinary expertise. So in current industry, most of the people who are working with the testing of cancer predictive things, they need the people who know the biotech or molecular biology as well as bioinformatics. Not only know how to use the bioinformatics, you can also develop codes or as well as develop database for this kind of research. Why we are seeking for such expertise, because if you are pure biotech students, you do not know much about bioinformatics. However, if you are pure bioinformaticians, you know a lot about programming, coding, but you can't link them with the biotech data. So this is a problem we face in current industry. So if you are looking for a job like bioinformaticians, like those interdisciplinary jobs, they give you high pay in the industry. So this is one of the things that we try to search for. Okay. So all of this thinking come with my mind with all 7 or 8 years of doing multi-omics and bioinformatics research. So I'm trying to do that whether I can contribute to society by using my expertise. So what I did for the first one is genetic consultation. So nowadays, you know, people talking about genetic testing. It's like, oh, you can predict certain disease, your risk of disease, as well as your nutritional needs. Yes, in scientific research is still ongoing, and is still, I would say, will be more and more accurate in future due to the advancement in technology. So my friend opened a company. So he actually also PhD graduates in biotech, also dealing with those medical and nutritional research. So he collaborated with Genome Institute of Singapore to come out with the genetic testing test, in which GIS from Genome Institute of Singapore, they actually help my friend to analyze those genetic testing data. Okay, for example, it's like the risk to get some cancer, okay, the risk to get some coronary heart disease. So it's predicted. Okay, so some of the tests that actually can predict potential talent and career. So it really looks like your destiny. Oh, you tend to become that person. It seems like that. But actually, what my role in my friend's company is that I will make sure those things not become destiny of certain people. It will only become some reference of your life. Okay. If, for example, if you have the risk inherited from your family, you will get high risk of diabetes, for example. So doesn't mean that you will get the diabetes. Okay, you can still live yourself well. For example, keep your healthy lifestyle. Okay, eat well. Then you don't have the risk. You will lower the risk. So this is what I did usually. Make sure the line does not cross. Make sure people when see their prediction, they won't feel like, oh, they're not going to live longer. So that is part of the work. Also provide consultation on scientific facts on that, right? So in this genetic testing, there are a lot of disclaimer that during the consultations, for example, you can explain in simple way, like what is mean by whole exome sequencing, usually deal with the rare disease. Okay, the single nucleotide polymorphism is usually for common disease. Okay, then it's important to mention that one of the risks or one trait, they are not controlled only by one gene. There are multiple gene control for certain trait or risk. So this is important as well as your lifestyle, your environmental factors, like how you live yourself in your daily life is important to influence. Okay, to influence your somehow inherited disease. That's also a good influencer somehow. All right, so psychological and ethical consideration, for example, we also like have a guideline to uh, protect the data, personal data, because the genetic testing is very personal and has high privacy. So we consider the ethical concerns as well here. So this is what part of my work, in the sense that not only can earn some extra money, but also helps to use my knowledge in contribute to the society. Okay, so I also shoot some videos. I'm not influencer yet, but I shoot some video helping my friends to clear the doubts on the genetic testing and ongoing technology, for example, NGS technology and some of the bioinformatics things. Aside from genetic consultant, I have another role. We call it Columnist. So my column name you can find in the China press in a Mandarin newspaper. Sorry for that, it's in the Mandarin newspaper. Okay, but I'm trying to write English right nowadays. All right, so Mandarin newspaper since 2020. My column name is named as Kepler. Kepler is a new planet. Okay, Kepler is similar to a word in Chinese mean make things simple. Okay, so simplified. So what my job in this column is actually simplify the complex scientific concept so that the general public can understand easily. For example, latest trend insights are like during COVID time we have a lot of Omicron, different variants, right? So I actually explain in the scientific content, like it's like you're fighting your ancestor, like that. Okay, like evolution, like using Pokemon as an icon to talk about evolution. That's very simple way, right? Okay, then link my own life philosophy to science or observation in life. So I think this week I published an article is related to a milk, how the milk spoiled it can be described as part of your life. So it's those things like make things that easier to understand, and people also learn what happens for certain scientific knowledge. As well as I share some of the experience as a student and as a lecturer. Sometimes we have some troubles and something to face as a lecturer. Any academic, for example, many KPI like very high. then, uh, then write it in a very, I would say, a very funny way, so that to tell people that's how's alive as a postgraduate student and a lecturer in academics. So those things actually help close the gaps between us and general public, because what we did in research is actually for the benefits of human being. So this is how I try to make the thing simple. Okay, you can find my column, just Google them, then you'll find it. Okay, so here are some of the examples, okay, shown here. So talk about ChatGPT is our friend, or it's not our, it's our enemy. So let's talk about that. Okay, talk about Pokemons related to evolutions. Talk about my Kazana journey as a Kazana scholar and alumni. Okay, talk about some of the food poisoning cases. Because I think I still remember last year with a case, people eat char kway teow and die from eating that because of the boric acid from one of the food pathogen. So we talk about that. Okay, how about food poisoning can be really a big case. Why and so? Those are the things that actually in my daily life, what I seen from the news, what I met. Okay, so I even write like how I work with Dr. Wang before the ICMBB, MSMBB works in the newspaper. So in a way, it's help to promote that, but it's win-win situations. Okay, so learn to communicate scientific knowledge in a simple way. And sometimes I critically evaluate certain scientific facts and data, and even environmental news and conservations. And doing some self-reflection. Luckily, I still have a good readers. Is respond well. It's always top 5 in the list weekly. So hopefully I can maintain that. Hopefully I can maintain that. Hopefully we still can maintain this thing. I have not been kicked out yet. Okay, still in the columnist area. Okay, hope I can maintain that in next few years. Okay, so one of the article has been chosen to become the content of Bahasa Cina Primary 6 textbook, one of the topics. So it's been chosen by MPM through the publisher. So I rewrite those things because when deal with the KPM, they have certain words they need to learn for that particular topic. So I need to incorporate those words in my writing. So I rewrite one of the article. Actually, this article is talk about metagenomics of dust. So how dust can cause airborne disease. It's actually, but I use a fairy tale way to talk about this. So I changed some of the way to write, then it become the content. So you can find it in primary 6 textbook. Okay, right. So with that, actually, my current three big careers are there. Hope it will be better and better. So of course, behind all this, there are a lot of friends, colleagues, and my students and my supervisor. It's a big team. So I have them. I'm very fortunate to have them from people from UK, people from UTM, and people now in Utah. Develop a small team. Okay, here. So and my postdoc as well. So they are helping me to have a better career. Okay, thanks to them, most of them. I don't think I'm perfect, but with them, it's make myself growth a lot, continuous growing. Okay. So thanks to all the sponsors here to fulfill all. I hope I fulfill their needs. Okay. All right. So thanks to them. So with that, this end of my presentation. Right. You can contact me well here. Thank you.
Okay, thank you very much, Assistant Professor Dr. Lam, for the interesting session and knowledge. I will begin the Q&A session now. If there is any question from the audiences here, you may ask. And also for the online participants, you may unmute yourselves and type your questions in the chat box.
All right, Dr. Lam, thank you for your very actually insightful, very interesting presentation. All right, so I think we have achieved quite a lot at a very quite young age. All right, okay. My first question actually is on the mangrove. Just now you mentioned that you're actually taking the soil sample from the mangrove. So where actually you get these samples? How can you ensure that this mangrove actually is similar to the different
Side of mangrove. Okay.
yes, because the population of the bacteria might be different, might be influenced by the surroundings. Maybe they have some, but whether it's I would say maybe the surrounding environment that factories.
Yes, it's fluctuating. It's fluctuating. We cannot control the fluctuation. So what we did is actually at that time I took 3 points in which there are 3 areas of mangrove. Which one is closer to terrestrial, another one is in the middle, the other one is close to the sea. So I took these 3 sites and make sure it's actually a few points at that area. So this is how we try to do, because my important thing is I don't do any diversity work. My purpose is to get their enzyme and protein. So sometimes that will be not my major concern. So I avoid that. Try to avoid that. Okay.
So,
Yes.
Sorry.
Yes.
Right?
Yes, soy sauce cake. Soy sauce cake food waste degradation.
Other than Angel brand, is there any other brand that using the soy sauce cake?
Yes, actually, because the process is similar. It's just that I'm not sure whether that industry want to declare that as a waste. Because not every industry they want tell them they have a lot of waste.
So you chose Angel because the brand declared that they didn't.
Yes, they in their SOP food certification, so they mentioned that this is only their waste.
Then my question, I think most of the postgraduate students are inspired by your presentation. And my question is how you divert yourself to multiple disciplines
Okay,
at one time.
I have a mindset that I'm not a very boring person, so I continuously seek for interesting works to do. Okay, so the important thing is I make myself flexible. So because people, if you are in certain area or certain niche of the research, you tend to be in there. Okay, but I don't want to do so because to me will be too boring. You can go very deep, it's true, but I try to make myself diverse in the sense that I can learn many things. I make myself happier, happier by seeing different work, doing different work.
So from your age, you already did what
Yes.
and then you
Yes.
explore more right after.
Yes. Yes. Because I was fortunate that my PhD time, that 3 years, is a Newton Fund project. So I need to work half in UTM, another half in UK. So it make myself flexible and widen my horizon actually. To see, oh, when I travel here, it's a lot of cultural difference. From there actually I open up myself. This is the first thing. And thanks to Kazana sponsor me all the time, then I can fly here and there to learn about the omics, because part of my job is actually to transfer back the things I learned from UK to Malaysia. This is one of the aim in the Newton Fund. But unfortunately, I don't have many chance to transfer those knowledge because when I back here, I graduated right before MCO. Right before 1 week before MCO, finished my PhD. So after that, it's a lot of things happen as we know. So the chance to transfer those technologies is not up to the standard yet. But I know it's growing because many people are realizing the importance of omics and they are focusing on that.
Thank you, Sharifah.
No courses. To be frank, mine is by chance. Okay, how I get into this industry is that one of my hobbies is to write posts in social media. So I have a connections who is an editor in the newspaper. So he found my writing interesting. So he developed a column, asked me to join whether I willing to do that. So the key answer is collection. Collections. So so I never thought that my writing can be so important that way. So it actually turns out to be. But I did few years lah, like 4 or 5 years writing of posts on Facebook, and then it's only in my own social account, not public one. So maybe the editor see some potential on it, so he invited me. So it worked until today.
Any other questions?
Dr. Lam, just now you mentioned that you actually write column every week. So I have been writing since
2020.
2020. So where do you get the inspiration?
Okay, the important thing is there's one thing I do every day is self-reflections. So I reflect myself since morning, the moment I open my eyes, what I did until before I sleep. So those give me a lot of inputs. Hey, am I doing right things? Am I appreciating what happens every day? Am I doing something that I can improve? So, and am I seeing something new in my daily life? So those things, when you accumulate together, it has a lot of ideas. I just need 2 hours to finish. Because the due date is every Tuesday before 12 a.m. so I will sit in front of the laptop in a coffee shop and just spend 2 hours and writing while listening some music and watching some videos. It's give me, I think that accumulation give me a good idea on what I need to write, what I need to focus, because we need to consider whether the reader interested or not. Because your column will stay there mostly due to your readers. So I have few thousand readers per article. So the statistics is quite good. I was lucky to be frank. I was lucky to be frank at that. So that actually I need to think what makes the thing interesting. What are the things that can catch people attention? That is part of my writing when I wrote some introduction or some ideas.
Okay, there is no question from the online audiences as well. So if there is no other questions, I will now end the session. Thank you very much to Assistant Professor Dr. Lam for the very engaging presentation. Thank you to the audiences here and also the online participants. Before we end our session today, we have a little gift as a token of appreciation for you that will be presented by our director. Before that, I will end the meet for the online participants. Thank you very much for joining. I will share the link to the attendance form at the chat box. you may fill in the form before leaving. Thank you very much.